Hello everyone. Today is a special day for all of us. So I would like to take this opportunity to wish all of you a happiest, healthiest, wonderful and a prosperous new year. So in our previous class, we have discussed regarding the mitosis. So we have undergone the various processes and we have learned that cell division is a progressive phase. So cell division takes place continuously. In our previous class we have learned. Now we are in the stage of anaphase. We have gone through various phases in the mitosis. Now today we have to learn in the early anaphase stage what is happening. You can see here in the picture, in the image, in the early anaphase what is happening? The centromere starts to split. The centromere starts to split. The chromosomes starts to move towards the poles. They are moving towards the pole. Pole की तरफ आधे इस तरफ और आधे इस तरफ. So half the chromosomes will be moving to that side and half the chromosomes will be moving to this side. So both the chromosomes are moving to the opposite poles. So in the early anaphase, the centromere split. Half the chromosomes move in one pole and half the chromosomes will be moving to the another pole. So in the early anaphase stage, the centromere split and the chromosome move towards the opposite poles. Next stage is late anaphase stage. Late anaphase stage, what is happening? In the late anaphase stage, the chromosomes have almost reached their respective poles. So the chromosomes are reaching their respective poles. In the early anaphase stage, it starts to move. Centromere split and it starts to move. And in late anaphase stage, the chromosomes reaches their respective poles. The cell membrane begins to pinch at the center. Here it starts to move. See here, the chromosomes are moving towards the opposite pole. The image is very clear to all of us. Next, let us move towards the telophase stage. And what is happening in the telophase stage? Let us learn. So telophase stage, we can say that it is the end stage of this mitosis. That is the end stage. You can see here the telophase phase. First we have seen the early anaphase, then we have seen late anaphase, and this is the telophase stage. So telophase stage, we can say that it is the end phase of the mitosis. So endoplasmic reticulum, Golgi complex, nuclear membrane, all these starts to form here. Okay, all these were disappeared. In the stage of prophase we have seen just opposite of prophase is taking place here. Okay, so here all the formation is taking place and telophase stage we can say that it is the end stage. So chromosomes again will start to condense. That means the opposite is of prophase is taking place here. So chromosomes may lose their individuality and opposite of the prophase, the reverse process of the prophase is taking place in the telophase. We have learned in the prophase stage what is happening for the endoplasmic reticulum, what is happening for the nucleus membrane and all. So here in the telophase stage we can say that it is the end phase of the mitosis. So nuclear membranes form around the appeared chromosomes here. Around the chromosomes nuclear membrane starts to appear and this is the end stage of the mitosis. So here the two daughter nuclei is being formed. Now moving towards the cytokinesis. So let us learn what is happening in the cytokinesis. So in cytokinesis, what is happening? In the hollow cell, the movement is taking place. You can see here the cytokinesis, the process of the cytokinesis. Here division is almost complete. Mitosis is over here. So mitosis is being completed. So one is here and another one is here. Two daughter cells are already formed. You can see the chromosome number also the same as in the parent cell. In the daughter cell also you can see the same number of the chromosomes. That's what we call it as equational division. Earlier also I have mentioned during when we have learned regarding the mitosis process. And in cytokinesis, what is important that you have to remember is that in animal cell how the cytokinesis is taking place due to the furrow methods. Through furrow methods it takes place in the animal cell. In plant cells, how it is taking place? Due to the cell plate formation. So in animal cells it is taking place through furrow method, and in the plant cells it is taking through the cell plate formation. Here in the cytokinesis, mitosis is, as I have told you, mitosis is completed. Two cells are being formed here. The same structure, same number of chromosomes we can see here as of the parent cell. Cytokinesis, that is an important point you have to remember. In whole of this process, what are the important points you have to remember? So in which condensation has been completed, in which phase the condensation has been completed. So these are the questions that has been asked from the beginning onwards. So which is the stage, that final stage, which stage is called as the final stage? We have learned that it is a telophase stage. And in which stage this endoplasmic reticulum, Golgi complex, nucleolus, nucleolar membrane, all these things appears. These also in prophase stage. Okay, so disappears. All these questions will be asked. Then which is the smallest phase? Which is the smallest phase? Anaphase stage is the smallest phase. And another question most probably asked is that in which centriole, in which phase it has gone to the move towards the centrioles has moved towards the pole. So we know that it is from the prophase stage, early prophase stage it started to move, and in which stage the chromosomes are in the equator, that also we have learned in the metaphase stage. Then we have learned that chromosomes, where the chromosomes poles are reaching in two different chromosomes are reaching in two poles. Okay, that is in the, and all these questions are always asked questions. So you have to remember here. We all know that after karyokinesis, cytokinesis is taking place. If it is not taking place, if the after karyokinesis, cytokinesis is not taking place, then there is a condition arising. What is that condition? Multinucleated condition will be arising there. So what we call it as? We call it as syncytium. Okay. For example, we can remember that is endosperm of the coconut. Syncytium is taking place in the endosperm of the coconut. Now let us move towards the further process. So here is the summary of what we have learned in the cell cycle and cell division. So in the mitosis process, various processes is interphase, cytokinesis, prophase, anaphase, in the telophase, as well as in the quiescent stage, what is taking place, G1 phase, S2 phase, and also the various phases, anaphase, early prophase, M phase, all these phases, what is happening, what is taking place, this is the summary here I have mentioned. Now moving forward, you can see, as I have told you, in the interphase stage. See, you can see here the cell membrane, centrosome, cytoplasm, nuclear membrane, chromatin, nucleolus, and in the early prophase stage, how the aster is being moving. Here it is very clearly given. In the metaphase stage, the chromosomes coming in the equatorial plane, and also in the early anaphase stage, how the centromeres are moving forward. All these are clearly given in this image. Next, here you can see in the late prophase stage how it is going to be formed two daughter cells, and here how the different stages of the mitosis in an animal cell. As I have told you, in the animal cell it is taking place through the furrow methods, and in the plant cell how it is taking place? It is taking place through the cell plate formation. Now moving towards the significance of the mitosis. So what are the significance of the mitosis? So the first point here I have mentioned is growth. The significance of the mitosis, the first point is growth. So we know that in every cell, growth is the important point. So in every cell, in every aspect, in every individuality, the growth is the important thing. So जितने भी somatic cells है, उसमें mitosis form होते हैं. Okay, how many somatic cells are there in that? Growth will be taking place. We know that mitosis is essential for the growth as well as for the development. Okay, mitosis is essential for the growth as well as for the development in the multicellular organisms. So जैसे ह्यूमन बॉडी है, लगभग कितने सेल होते हैं इसमें? 6 into the 10 raised to the power of 12. ओके, इतने सेल्स होते हैं सिंगल बेबी में. 6 into the 10 raised to the power of 12 सेल्स होते हैं सिंगल बेबी में. ये कैसे बने? हाउ इट इज बीइंग फॉर्म्ड? इट इज बीइंग फॉर्म्ड फ्रॉम अ सिंगल सेल, zygote. ओके, zygote से ही बने होते हैं all these things. So next, moving towards the cell repair. So what is happening in the cell repair? So where this cell repair is taking place? Where this cell repair is taking place? So they are providing a significant contribution. So cell repair provides a significant contribution. जैसे cell layer का upper layer जो है, epidermis, we call it as epidermis, or we can say that the lining of the gut, or we can say that the blood cells are constantly replaced. Okay, old cells are being replaced. If there is any injury occurs, then what happens? All the healthier cells surrounding that cells will be going and setting there. Okay, so what happens? Surrounding healthy cells में cells से ये भर जाती है. गाँव जो बन जाती है वो सराउंडिंग हेल्थी सेल से भर जाती है. So the injuries will be replaced. How the cell repairing is taking place. The next significance of the mitosis is reproduction. So what is happening in the reproduction? In this mitosis process, multiplication is taking place. So organism is being multiplied. The organism is being multiplied. So another significance is reproduction. And the next significance is maintenance. As I have told you, the next significant division is maintenance. What is happening in the maintenance? We know that in mitosis, the replication will be taking place. So what is happening? Equal division will be taking place. Equal division is taking place. तभी तो मल्टीसेलुलर ऑर्गेनिज्म का नंबर और टाइप ये सब कुछ सेम रह जाता है. Then only it will be of same. The parent cell, the chromosomes number, the type, everything will be of same. That's what we call it as equational division. The next important significance is that it restores the nucleocytoplasmic ratio. So any of the efficient cells में ये जो nuclear cytoplasmic ratio will be more. Any of the cells में efficient cell का ज़्यादा रहता है क्या? Cytoplasmic ratio. तो what will happen? Size will be increased here. So if the size is increased, then what happens? The ratio will be decreased. So इसको सही लेवल पे लाने के लिए मैकेनिज्म हो रहे हैं और एफिशिएंट लेवल पे लाने के लिए इसमें मैकेनिज्म हो रहे हैं. सो न्यूक्लियोसाइटोप्लाज्मिक रेशियो के लिए ये डिवीजन चलते हैं माइटोसिस में. अंडरस्टूड? सो नेक्स्ट इज ऑफ कैंसर. Cancer, we know that in the mitosis division, it is going in an uncontrollable level. The last significance is cancer. So in the mitotic division is uncontrolled. This mitotic division is becoming uncontrolled. So that is going to the cancer. That means uncontrollable divisions. So cell division is being replicated or duplicated, we can say. So इसमें कैसे ग्रोथ हो रहे हैं? जैसे एपिकल मेरिस्टम, लैटरल कैम्बियम, इसमें कैसे ये इस सारे में कैसे ग्रोथ हो रहे हैं? सो इस सारे ग्रोथ जो है माइटोसिस डिवीजन के द्वारा ही है. So I hope you might have understood the significance. So the first point what we have learned was growth, and the second point we have learned was cell repair, how the repairing is taking place when the injuries occurs, healing, then the reproduction. Next is maintenance of the chromosome number, how the chromosome numbers are maintained as in the parent cell. Then the next is restores the nucleocytoplasmic ratio, and the last point we have learned is cancer. So that's all regarding the mitosis. From the beginning onwards we have learned what is mitosis, what are the various phases of the mitosis, why it is called equatorial division, and which is the dramatic cycle of this mitosis. All these things we have learned in details. So please go through the notes and prepare a notes. Thank you. May God bless you all.